Um, and another thing that's come out very recently is a paper from Dr. Gao-Ling Ming's group at University of Pennsylvania, and they are using cell models. So a few years ago, we started making some investments in stem cell research for PWS as a way to better understand PWS. And it's really nice to now see the fruition of some of those investments, and this is one of them. So this group is recapitulating sort of a mini brain in a dish. And they've been able to develop a technique to take stem cells and kind of turn them into a part of the hypothalamus in a dish. So the arcuate nucleus is the particular part of the hypothalamus that's kind of critical to hunger and satiety. So it has all of the neurons that regulate those hunger and satiety properties. So these investigators figured out how to take the stem cells and grow them over the course of 70 days into a little ball that represents the arcuate nucleus of the hypothalamus. And then they did that with FPWR funding with both typical neurons and then the PWS neurons. And then they asked, okay, so what's different? And they found a lot of things different, actually. So firstly, they found that there were more precursor cells in the PWS organoids, indicating that there may be a problem with so cells grow and then they kind of differentiate and do their business. And what they found was the PWS cells, they grew, they were slower to differentiate and kind of do their jobs. And in this case, the job is regulating that hunger and satiety. They also found that they had decreased responsiveness to leptin. Leptin's a hormone that tells your body that you've got plenty of fat, you don't need to eat anymore. And these organoids, even separated from a person, were not very responsive to leptin. So that's an important feature that is important to understand and consider as we develop new drugs for PWS. And they showed, and this kind of built on the work of others, that there is increased microglia or inflammatory cell infiltration when they grow the organoids in certain conditions. And this suggests potentially a new therapeutic pathway, that maybe there's an inflammatory component that is sort of over robust in PWS compared to typical individuals. And so this paper really builds on our growing knowledge of how the brain and brain function is different in PWS. And that's important for understanding what new targets for therapy might be, and also gives a potential model in which you could test and see if particular drugs kind of correct things and bring them back more towards the way typical neurons behave.